Welcome to the October 2021 in-service for the Kent Northwest Kidney Center, the greatest dialysis unit in the history of modern medicine and the flagship unit for the Northwest Kidney Center. This month's topic is influenza. It's one of our mandatory annual topics, so you've heard it before and you'll hear it again. This is a very important topic because influenza has just as much potential as COVID to cause a pandemic and widespread illness and death. There are four objectives to today's talk: understand the microbiology and the clinical impact of influenza, recognize its clinical presentation, appreciate vaccine history regarding influenza, and finally be familiar with how influenza vaccine is produced. Okay, let's start with some general background regarding influenza. Infections caused by influenza fall into one of three categories. They can be local infections, or they can be an epidemic or a widespread occurrence of an infectious disease in a community at a particular time, or they can be a pandemic, which is prevalent over a whole country or the world, a term that is very familiar to everyone courtesy of COVID-19. Influenza pandemics are believed to occur every 2 to 3 times per century, although there have been 5 major global pandemics since 1900. There was the 1918 Spanish flu, or the H1N1 vaccine, which killed estimated 50 to 100 million people worldwide, or 3 to 5% of the world's population. There was the 1957 Asian influenza, H2N2, which killed about 1.1 million people worldwide and 116,000 people in the United States. The 1968 Hong Kong flu, which killed more than 1 people worldwide. The 1977 Russian flu, which killed about 700,000 people worldwide. And finally, most of you probably remember the 2009 Swine Flu Pandemic, also an H1N1 virus, just like the Spanish flu, which killed about 284,000 people worldwide. Of all these pandemics, the Spanish flu was the deadliest. It killed more people in 24 weeks than AIDS did in 24 years. Killed more people in 1 year than smallpox or the Black Plague did in 50 years. Killed more Americans in 1 year than died in combination World War I, World War II, the Korean War, and the Vietnam War. And finally, it killed 2 to 20% of those infected compared to a typical annual mortality rate of only 0.1%. During the Spanish flu at the University of Michigan at Ann Arbor, any student caught without a mask during the Spanish flu was automatically suspended. And a town in Arizona passed a law forbidding people to shake hands. Now, if I had told you this before the COVID-19 pandemic, many of you would have been shocked. But now that we are living through the COVID-19 pandemic, this does not seem unusual or surprising. Let's now talk about the microbiology of influenza. The term influenza comes from the Italian word meaning influence, and it refers to the cause of the disease. It was felt secondary to unfavorable astrological influences such as the planets and the stars and the moon. The English adopted the word influenza in the mid-18th century. The French called it la grippe, from gripper meaning to grasp or hook, because the illness grasped a hold of you and refused to let go. In Arabic, the term influenza, which actually sounds a lot like influenza, means nose of the goat, and it's used because goats were correctly thought to be carriers of the disease. Influenza is a disease of birds and mammals, so pigs, chickens, other birds, and people all can pass influenza from one population to the other. Influenza is a family of RNA virus, actually just like coronavirus. Influenza viruses have a negative sense single-strand RNA genome. The negative sense of genome means that it can be used as a template to synthesize messenger RNA or mRNA. There are two main types that cause human disease, influenza A and B. Influenza B is less common and almost exclusively affects humans. There is actually an influenza C as well, which causes very mild illness among humans, and there's an influenza D, which predominantly affects cattle but is not known to affect humans. Influenza can be identified by two types of surface proteins. One is called hemagglutinin, and the other one is neuraminidase. These are, again, two different proteins that define the type of virus. And so that's why you oftentimes hear the strain referred to in terms of these letters. So the Spanish flu was H1N1, Asian flu H2N2, Hong Kong flu H3N2, the bird flu H5N1. And that's because the virus is constantly changing. It's rearranging the types of proteins on its cell surface, which allows it to evade the immune system of hosts. So how does the influenza virus keep changing from season to season? Well, there are two mechanisms. One is mutations. So basically, you can get mutations in the genes that encode for hemagglutinin and neuraminidase antigens on the surface of the virus. That's called antigenic drift. More commonly, we see what's called reassortment. Okay, where you get, let's say one bird gets infected with two different strains of influenza. Well, the virus then enters the same cell or cells within that bird where the proteins and the mRNA can get packaged differently. So basically you can create a whole new pathogenic strain as a combination of two different viruses that are co-infecting the same organism. Now let's talk about the clinical presentation of influenza. Hippocrates was a Greek physician in classical Greece who is considered to be the father of medicine, basically in recognition of lasting contributions to the field, such as the use of prognosis and clinical observation. He also systemically categorized diseases, and he came up with something called the humoral theory. But among his contributions to medicine was a description in 412 BC of the influenza outbreak that occurred 2,400 years ago. He described this influenza-like illness, which he called fever of Perinthus, that many scholars claim is probably the first historical description of influenza. So what are the clinical features of influenza infection? Most of you are very familiar with these, and these include chills and fever and sore throat. A lot of people have muscle aches, headache, coughing, weakness, fatigue, general discomfort, nausea and vomiting, etc. These symptoms are often sudden, and that's why influenza is also called the hit by a truck disease. Symptoms typically begin 1 to 2 days after exposure to the virus and last for about 2 to 8 days. Diarrhea can occur, vomiting, particularly among children. Complications of influenza include pneumonia, which can be caused by the virus or by a secondary bacterial infection. Other complications, potential complications include tons of inflammation in the lung, which can lead to something called acute respiratory distress syndrome. We see that in COVID as well. People can get meningitis. They can get a viral infection to the brain called encephalitis. And of course, if you get really sick with influenza, it can exacerbate any other health conditions that someone has, like asthma or heart disease. Most of you are also very familiar with the transmission of influenza. Basically, it's transmitted through coughing, sneezing, which basically generate lots of aerosolized droplets containing the virus. And influenza can survive on hard surfaces for days. And of course, just like with COVID, airplanes are a wonderful way to enable the virus to travel internationally in a matter of hours, and there have been outbreaks of influenza reported aboard commercial airliners, just as with COVID. In a typical year, 5 to 15% of the population contracts influenza. There are usually 3 to 5 million severe cases of influenza annually, typically 650,000 respiratory-related deaths globally each year. The deaths occur most commonly among high-risk groups, including young children and the elderly, but also people with chronic health conditions such as end-stage renal disease. Cases peak in winter in temperate areas, but occur year-round in tropical climates. The best way to handle influenza, of course, is prevention. As with the COVID pandemic, there are two ways to prevent influenza infection. One is with proper hygiene, and the second is with vaccination. As an aside, during the 1918-19 Spanish flu pandemic, people didn't know the cause of the infection. They didn't know that there was an influenza virus that hadn't been discovered yet. They didn't know if it was secondary to bacteria or if it was a dietary deficiency. So people came up with all sorts of ways to prevent influenza, and one of them was to eat more onions. And in theory, if eating an onion gave you really bad breath, such that you were less likely to be kissed by your spouse, that could work. Of course, working in a dialysis unit, you all are experts with proper hygiene. But just for review, this includes washing your hands, covering your mouth if you cough or sneeze, avoiding close contact with sick people, staying at home if you're sick, and of course sterilizing surfaces with, say, diluted chlorine bleach. Of course, the second way to prevent influenza infection is with vaccination, and we are in the midst of flu shot season. There are two types of flu shots currently available. One is a trivalent vaccine, which protects against three strains of the flu. They have two influenza A strains and one B strain. More often we use the quadrivalent vaccines, which protect you from 4 flu strains, have 2 influenza A strains and 2 influenza B strains. This is becoming more popular over time. This is what we have at our dialysis unit. Influenza vaccination is, of course, recommended for almost everyone, particularly kids, seniors, those with chronic disease, including end-stage renal disease, of course, those who are immunosuppressed, such as transplant recipients, their household contacts, people who live in crowded environments, people who go to school, for example, or live in dormitories or work in a crowded building. All healthcare workers are recommended to be vaccinated, and pregnant women as well. In addition to trivalent and quadrivalent vaccines, we have what are called high-dose vaccines. These have been approved for use in the United States since 2009. They're designed specifically for people aged 65 and older, as well as people who are relatively immunosuppressed, and they contain 4 times the amount of antigen or protein as the regular flu shot. And this is associated with a stronger immune response following vaccination, allowing for higher antibody production. So why should people greater than 65 receive the high-dose flu vaccines? Well, elderly amount less of an immune response to vaccines. That's why they're also encouraged to receive a COVID booster shot. There was a study published in 2014 which looked at 30,000 people above the age of 65 and showed that those who received the high-dose vaccine had 24% fewer influenza infections compared to those who received the standard dose flu vaccine. Typical side effects of the flu vaccine include pain at the injection site. Kids oftentimes get a fever, but that's rare in older children and adults. And sometimes people get a runny nose or a sore throat. There is a possible association between the influenza vaccination and something called Guillain-Barre Syndrome. Guillain-Barre is a form of ascending paralysis, starts at your toes and spreads all the way up your body, that has been previously thought to occur in the frequency of 1 case per million doses. But the CDC says that modern vaccines do not appear to have any association with Guillain-Barre. And a large study in China reported in the New England Journal of Medicine looking at over 100 million doses of vaccine against the 2009 H1N1 swine flu, found only 11 cases of Guillain-Barre, so 0.1 per million dose. It's actually lower than the normal rate of Guillain-Barre in the population. So it seems likely that Guillain-Barre was associated with previous preparations of influenza vaccines, specifically in the 1970s, but not with current preparations. On average, the influenza vaccines are about 50 to 60% effective in preventing disease. This is pretty good, but it pales in comparison to the effectiveness of the mRNA COVID vaccines. A couple of myths regarding the influenza vaccine. One, I could get the flu from the injectable flu vaccination. So the virus particles used are killed and not infectious, so there's no risk of transmitting disease. It has very low reactivity. So some people might develop body aches and chills and things, but that's uncommon. That said, the intranasal preparation of the flu vaccine, so the non-injectable nasal spray, is a live virus. So people in theory could get infection from the nasal spray. Second myth, if I have an organ transplant, flu vaccine could be harmful. That is not true. Flu vaccination is recommended for all transplant patients. Contraindications to receiving the injectable influenza vaccine include a severe egg allergy, that is if eggs were used to make the vaccine, a latex allergy, current moderate severe illness. So if you have a fever or some other infection, we would wait until that resolves before we give you the influenza shot. And finally, if you have a history of Guillain-Barre syndrome, which again is that paralyzing illness, again, the association between that and influenza vaccination is very, very weak. But it's such a horrible, medical condition that we don't take the chance. So now I'd like to spend a little bit of time on influenza vaccine history. And this is just for fun. I don't know if I mentioned this previously, but I majored in history in college, and history to me is just a lot of fun. As I mentioned previously, during the 1918-19 Spanish flu pandemic, no one knew what the cause was. Some scientists began to suspect that bacteria were not the real agent of influenza disease, but they didn't know that it was a virus until the 1930s, when in 1932, a few English scientists, I think their names were like Wilson Smith, Christopher Andrews, Sir Patrick Laidlaw. Basically, they were working at the Medical Research Council. They first isolated influenza A virus from the nasal secretions of infected patients, and this essentially demonstrated the intranasal human transmission of the virus. Then in 1934, the American virologist Thomas Francis Jr., working with Smith in England, was able to transmit the virus to mice. In 1935, a guy named Sir Franklin McFarlane Mrna and Smith separately discovered that the flu virus could be grown in hen eggs, a procedure that is still used today to manufacture most influenza vaccines. In 1936, the first neutralizing antibodies were generated by an infection and isolated. 1937, there was a study among military recruits in England used a subcutaneous vaccination with an inactive strain of influenza A, so the first time that humans had been successfully vaccinated against influenza. And in 1938, Jonas Salk, who then went on to develop the polio virus, worked with another guy and did the same among U.S. military recruits. So in a very short period of time, they identified the virus, they demonstrated its transmissibility, and they came up with a vaccine, all within less than a 10-year span. In 1940, influenza B was discovered and subsequently shown to be an important cause of seasonal epidemics and subsequently an important part of annual vaccine programs. In 1942, large studies were conducted on the first influenza virus vaccines in the general population, and they provided the first official proof that an inactivated influenza vaccine could be protective against a flu epidemic or pandemic. In December of 1942, the first bivalent vaccine. So currently we have trivalent and quadrivalent vaccines, but in 1942 they introduced the first bivalent vaccine containing viruses of type A and type B. In the 1950s, the World Health Organization created the first system for the surveillance of circulating influenza virus strains, and that continues today. This is how we know what's circulating in the southern hemisphere and the northern hemisphere, and is an important part in the determination of the antigens that are used in the annual vaccine program. In 1968, there was a pandemic that led to the development of the first trivalent inactivated vaccines against influenza viruses. And the first flu vaccines were licensed in Europe, and annual influenza vaccination was recommended in the U.S. for those at major risk for influenza complication. So up to this point, all the influenza vaccines that had been produced were called split vaccines. Basically, they took a virus and they treated it with some sort of detergent that broke it up into all sorts of miscellaneous pieces. But in the 1970s, they came up with the idea of subunit vaccines. So rather than just totally destroy the virus, they basically broke it into subunits. So they broke it into the hemagglutinin and the neuraminidase components. And they found out that those generated a better immunogenic response. So people responded better to those. So in 1976, the first subunit vaccine was developed, and it contained only the surface antigens, hemagglutinin, and neuraminidase. And that's what we do today. Very immunogenic, very well tolerated. In 1980, the first subunit vaccine was licensed in the United Kingdom and continues to be available worldwide. And the composition of the vaccine started to be updated every year. So basically, for the last 40 years now, we've been changing the composition of the influenza vaccine on an annual basis. That, of course, requires surveillance systems. All right, let's now talk about vaccine production. How are vaccines produced? There are three main approaches to developing an influenza vaccine. One uses eggs. So it's an egg-based flu vaccine. That's where most of our influenza vaccination comes from. There's also a cell-based flu vaccine, which has some advantages over an egg-based approach. And coming soon to you is an mRNA influenza vaccine. These are currently in trials. All of these are made by commercial companies. The use of eggs is the most common way of generating an influenza vaccine. That's been used for more than 70 years. It's used to make the inactivated or killed vaccines, as well as the live attenuated or weakened vaccines that are used in the nasal spray. Eggs are fertilized and they're typically 11 to 12 days old. So the first step in making an egg-based flu vaccine is for the CDC or the World Health Organization to identify the candidate vaccine viruses, so whatever they think is circulating in the population at that time. And then those candidate viruses are injected into the fertilized hen's egg and incubated for 48 hours at 37 degrees. The egg is then cracked open. You get about 10 milliliters of fluid, generates about 15 micrograms of flu vaccine, and that's about one dose. So one egg gives you basically one vaccine dose. The virus is killed or weakened, and the viral antigen is purified and collected. It's an efficient system, but it has some shortcomings. I mean, you have to order the eggs 6+ months in advance. Again, you need one egg per vaccine dose. So you want to vaccinate 200 million people, that's a lot of eggs that you need. It's approximately 200 million eggs. And then, of course, you have to be aware of the fact that the chickens or the embryos could be killed if you happen to have a particularly virulent bird flu. And let's say you have a pandemic. I mean, it's really hard to upregulate the production of eggs to meet the demands of a population. So, if there were a pandemic and you needed to generate more flu vaccine, we would then turn to cell-based flu vaccines. Basically, these are vaccines that are produced by animal cells. It involves sort of a closed-loop bioreactor in place of millions of eggs, and it's produced not with chicken eggs, but with animal cells, specifically using something called recombinant DNA technology. And this involves inserting the the RNA of the virus, encoding for the antigen, like the neuraminidase or the hemagglutinin protein, that stimulates an immune response into bacterial or mammalian cells, and then they express the antigen in these cells, and then we can purify the antigen produced by them. So basically, there's a lot of potential for faster startup of the flu manufacturing process. So the steps involved are pretty similar to an egg-based approach. Again, the World Health Organization or the CDC identifies the candidate virus for vaccine. And then the manufacturers inoculate those viruses into cultured mammalian cells rather than eggs and allow them to replicate for a few days. Those mammalian cells then generate the flu proteins, so the neuraminidase and the hemagglutinin, and that virus fluid is contained and the antigens are purified. And then after testing and FDA approval, it's released. In the not so distant future, we will also have mRNA influenza vaccines, much like the COVID vaccines produced by Moderna and Pfizer. So messenger RNA vaccines, or just mRNA vaccines, again contain the genetic code for the hemagglutinin and neuraminidase proteins on the influenza virus surface, and they are directly, so the mRNA is directly injected into humans rather than cells in a lab, and your own cells end up becoming a factory for your own vaccine. It gets injected intramuscularly, so the first cells to start produce the proteins are muscle cells, but then the mRNA gets delivered to multiple types of cells, including what are called antigen-presenting cells, which are very efficient at generating these viral proteins. Advantages are they're really fast to make. You don't have to go through a long process of producing and purifying protein in culture for later injection. They're probably more immunogenic, as exemplified by the fact that the vaccine efficacy is much greater, at least with regards to the COVID vaccine for the mRNA vaccines compared to, say, the other recombinant old-fashioned vaccines. Takes only weeks to design and test, so you can generate an mRNA vaccine very, very quickly. And several companies, including Moderna and Sanofi, are currently have mRNA vaccines in human trials. So this will be something available to us in the not so distant future. So just a brief word on treatment. If hygiene and vaccination don't prevent the disease, someone gets influenza, what can we do? There are a few medicines available. The most commonly used one is called Tamiflu or oseltamivir. It is a neuraminidase inhibitor. We actually have a standing order for treatment for flu at Northwest Kidney Center. If a patient has fever, plus runny nose or stuffed up nose or a sore throat or a cough, we can, of course, obtain blood culture that's febrile and then ask the nephrologist to prescribe Tamiflu. These days, it's a little bit more complicated, and probably the standing order needs to be revised because people could also have COVID, and so probably first they need to be tested for COVID, and at the same time they could also be tested for influenza, and then just empirically start on Tamiflu if the suspicion for that is high. So that's it for all the new information. Just in conclusion, remember that influenza can have a devastating worldwide impact. Symptoms often begin suddenly and include fever, cough, muscle aches, fatigue, so you're hit by a truck disease. You can prevent the flu with proper hygiene and vaccination. Administer Tamiflu if you suspect a dialysis patient has the flu.